All right, welcome. So, this is an overview of this week's lab. We're going to be talking about blood. So, blood, these are the learning objectives. Describe the components, that is, the formed elements and plasma. So, we talk about different formed elements and the physical properties. So, some physical properties that we are going to talk about here are the hematocrit, for example, which is the proportion of red blood cells in a blood sample. The then we'll also talk about the I want to call them the immunological properties. You can think of them as chemical properties of blood as well. So we'll talk about antigens that are on your blood cells and talk about proteins that are part of the soluble part of blood that are called globulins, immunoglobulins that respond to the I'll call them carbohydrates or markers, or better word, antigens on the red blood cells, and how that leads to the process of agglutination. So you will actually do the hematocrit in lab as well as you will do some agglutination reactions to determine the blood type of unknown individuals. Exciting, right? So first off, the most common blood type in the US is O positive. So it's depending on your race and ethnicity, could be as high as 53%, half of certain ethnicities. So first so what is agglutination? So I talked about red blood cells that have antigens or markers on them. So this first column, A, has A type antigens on it. So those tiny little circles are A antigens. And in antibodies are a type of protein in the plasma that recognizes specific antigens. So it doesn't make sense for me, for my antibodies, to target for destruction antigens that are on my own cell. If you had that, you would say you have an autoimmune disorder, auto self immune disorder. And so those are antibodies. They are in plasma. They're proteins, soluble proteins that will target the markers or the antigens on red blood cells. And next you have um, the blood type here. So there's also another type of antigen that is on red blood cells that it's not shown here. It's called the rhesus antigen. But I'd like you to watch this video if you can. It talks about the agglutination reaction. What exactly is happening? Basically, it's antibodies attacking antigens to the point where they form a clump together, and that clump is actually visible. We will use that principle to identify the types of antigen or the blood type of an individual. There is a practice resource. So the question here is who can I donate blood to? Who can receive my blood and who can give me blood? So there's two different resources that I'd like you to practice with. Again, scan this QR code and uh, for yourself figure out who you can give or who you can receive from. Now, there's I talked about blood containing formed elements and plasma. So the formed elements comprise your cells or platelets. So for the cells, there's white blood cells, which are red blood cells at the bottom, which are the most abundant, about 6 million per microliter. And then you have the leukocytes and then the platelets. Now, we'll talk about the different types of leukocytes. Eventually, you do that through APR. But in terms of abundance, it's the leukocytes, the white cells. It goes from neutrophils to lymphocytes to uh, mast cells, the monocytes, eosinophils, and basophils. And the acronym for this is Never Let Monkeys Eat Bananas. So The most abundant are your neutrophils, and the neutrophils are the ones that have multiple lobes. That's right here um, in the figure, and we'll describe a little bit more in lab. Then we'll talk about factors that influence um, the your red blood cell density or your hematocrit. And for the fun of it, I'd like you to watch this video and just watch the first 3 minutes. You don't need to watch everything. And tell me some factors that influence um, hematocrit and why that might be important. Um, the question I would ask you is if I took my own blood and I give it back to myself, am I doping or not? So you can watch that if you have some time.